Hi everyone, my name is Alexander Scala, and today I will be presenting the paper, The Parkinson's Disease Drug Entacapone Disrupts Gut Microbiome Homeostasis via Iron Sequencing. It's been published in Nature. It was also published in 2024, which is relatively recent, and the first author is Pereira. I now would like to go into the background of the paper as well as the authors and researchers' inspiration to the paper, including previous relevant publications, the demographics of the patients affected by such issue, as well as how many patients are currently using these two drugs that I'm going to mention later on. In the paper's introduction, the following paper, Extensive Impact of Non-Antibiotic Drugs on Human Gut Bacteria, is quickly mentioned. The author mentions it as a landmark study in which it examined 1,000 currently FDA-approved drugs and their impact on 4 gut bacterial strands that the authors determined is representative of our gut microbiome. The study is really important as it elucidates that 24% of drugs in the study were proven to show to inhibit bacterial growth. So we talk about a lot of adverse effects of certain therapeutics, but I feel that the gut microbiome changes and composition are highly overlooked, and this is showing to be an area of research that is further understood, especially within the realms of mental diseases, especially depression. The gut microbiome and changes to such composition are proving to be a lot more important than scientists have predicted. All in all, the authors were inspired from this landmark paper to conduct how 2 very important psychiatric drugs used for schizophrenia and Parkinson's disorder may have similar effects to those 24% of drugs that inhibit the bacterial growth. The paper I will be presenting focuses on 2 main drugs, entacapone, which is a Parkinson's disease disorder drug, and loxapine, which is used for schizophrenia. So on the left, we have the chemical structure of ET, and for our purposes of the presentation, I'm going to be referring it to as ENT and LOX. LOX is the schizophrenia drug, and Parkinson's disease is treated with ETN. The bottom line of this paper is that it is a study examining how these 2 nervous system targeting drugs impact our gut microbiome on a taxonomic and cellular level. So it's going from a very broad perspective to also examining the metabolic activities of single cells. As we find in this paper, high concentrations of ENT are going to be the most problematic for the gut microbiome, as well as LOX is going to display lesser impacts, but also significantly changing the composition and cellular activities of the bacteria in our guts. Particularly, they both led to the decrease in abundance of bacteroids and Clostridium sensostricta 1. ENT had a greater taxonomic effect compared to LOX, and that's in examining ENT low concentrations. So when I refer to ENT low versus ENT high, we're going to first discuss that that's just referring to the concentrations the patients were exposed to. And these taxonomic changes and perspectives data were acquired by long-read metagenomic sequencing of the fecal samples, and we're going to go more into sequence sample information later on. As mentioned previously, ENT and LOX both significantly reduced microbial activity at the single cell level, impacting, and displaying the author's variety in this paper, talking about a high perspective and then a very specific perspective. ENT is going to be further examined in the paper as it is bioaccumulated in certain bacteria, mainly E. coli, in which it also complexes iron in certain bacteria, mainly E. coli. This paper was published at an extremely hot time for mental disorder related therapeutics, as well as the current directions of literature are aligned with what this paper discusses. First, would like to say, why is this paper important and why are we examining Parkinson's disease as well as schizophrenia? Well, combined, LOX and ETN are produced in over 30 million tablets each year and distributed in the United States alone. So we have a high amount of these drugs being distributed to many different patients, many different demographics all across the country. As current literature kind of unfolds, there is a more pressing importance to the gut microbiome in relation to different disorders, whether that be depression, schizophrenia, or Parkinson's disease. So we are examining that, hey, changes, large global shifts in the microbial composition can either worsen or lessen symptoms for these patients. So combined with that, there is a very, very important need to study, are these drugs making our gut microbiomes worse or for the better? Not to mention, FDA, as well as more and more research, is suggesting that neuropsychiatric drugs are entering the U.S. drug market and becoming approved. As these neuropsychiatric drugs are entering in combination with the fact that more mental health related research is considering the microbiome, we need to understand how the drug microbiome interactions help or worsen these therapeutics, as well as are they harming our own gut. Now that we established the importance as well as the need to examine the drug microbiome interactions, I'm going to explore the study design and methods. The way I'm going to present the following information is based on the figures, the 6 significant figures mentioned and presented in this study. Each figure has its own relevant procedure, and we're going to break it down like that. The paper is broken down into 2 following questions. How does these 2 neuropsychiatric drugs influence the gut microbiome on the global level, but also on the cellular level, and what mechanisms are relevant to either iron starvation or inhibiting the growth of other neighboring bacteria? Each figure relates to the following. Figure 1 is important for understanding global microbial community composition. Figure 2 is important for understanding single cell activity and how are ENT and blocks impacting or harming the metabolic activity of those cells. Figure 3 elucidates how ENT is bioaccumulated in certain cells, while figure 4 goes into the iron chelation and starvation strategies of ENT in particular cells. Lastly, we examine how E. coli is actually promoted, growth is promoted via certain strategies, including iron scavenging, and how ENT kind of exacerbates that process. All the sequencing strategies in the paper are based on one source of sample prep, in which in this right corner where we're going to examine figure 1A, we see 6 fecal samples were collected from 6 healthy individuals. And these were varying in different concentrations, either ENT high, which represents a patient with a higher dosage of ENT, or ENT low, and then LOX low and LOX high. Once these were collected, they were placed in an anaerobic tent for 30 minutes post-sampling, and then suspended in said medium in order to gain a specific concentration of the fecal matter. Then the samples were aliquoted, pelleted, and stored at -80 for metagenomic analysis. However, all the supernatants from the previous steps were collected and pooled together and either placed in a SM9 median or a brain-heart infusion median. As we see, the control for the future experiments, it include DSMO, and the other variables being tested are the impacts of ENT and LOX. And as we get into, incubation is an ex vivo, and it occurs either for 6 or 24 hours, and that will be examined later on. And these times will be referenced and compared to each other as we move on. And B is evidence that there is cell growth throughout the incubation process, and that they have adequately prepared and incubated the cells for future experiments. The main question asked in figure 1, C, and D is, how does ENT and LOX affect the overall global composition of these samples? The main point of analysis is 16S rRNA gene amplicon analysis, and the first step with that would include PCR amplification, in which they use a two-step barcoding PCR. They first had to purify and normalize all the data, the samples, which was acquired using Illumina TrueSec NanoKit. Um, as to for completing the actual sequencing, it was sequenced on Illumina Minisec with 4 negative controls per 90 samples, and then the following sequences were deposited in NCBI sRNA. As they processed the raw data that was completed using FastQ and the DADA2 package in order to generate and then process the amplicon sequence variants, which are going to be proven to be very useful throughout this paper. And the long read sequencing was acquired through Oxford Nanopore. Figure 1 also includes a metagenomics heat map that displays changes to absolute and relative abundances of the microbial composition, in which this is accomplished using the data from the Illumina and Nanopore reads, but mapping it with Minimap 2 instead. The assembly is used with MetaBait2 in order to develop the circular contigs separately. Specifically, looking for AMR-related genes, they used FE-Genie for iron-related genes, and then AMR Finder Plus for those antimicrobial resistant genes. This will be proven really important for the last figure, in which the study examines how these changes in composition actually promote the encoding of antimicrobial-related genes. Figure 2 examines the single-cell metabolic activity of the bacteria, in which, when during incubation and after, they were exposed to D2O, which is just heavy water, and this was used to track the drug-induced changes in the activity. These changes were detected by stimulated Raman scattering, in combination with fish, which is a proprietary method that they previously have shown and proven successfully to determine gut microbiome responses to sugar in a high throughput manner with increased sensitivity. So this is an extremely interesting point in the paper where they're applying a previous proprietary method to this application. And then SRS and FISH was used for high throughput analysis, in which 8,000 cells were read at the 6-hour and the 24-hour mark. It is important to note at this point in the study that the ENT high signals, it was full of a lot of a noise, and it is excluded from the activity measurement. So the only data that will pertain to the single cell metabolic activity is ENT low, LOX high, and LOX low. After observing a lot of noise in the SRS fish signal for ETN high, the authors wanted to explore why this was the case. In turn, they determined that ENT was bioaccumulated within certain bacteria. So they wanted to prove it, in which they had to use a 10 micromolar concentration of ETN solution in order to develop a extremely strong photothermal signal. And this was used with picosecond SRS detection, and this was subsequently proven throughout the paper, as we're going to examine in the fluorescence images later on in the figure 3. This is an extremely interesting point in the paper because not all bacteria display this ability, and this is going to be proven in order to determine how ENT is retained, as well as also diminishes the surrounding nutrients for the cells. Figure 5 is particularly important because it is where the author and researchers provide extra iron within the incubation, and the cells react very differently to that. So it is supplementing iron to them, in which they use the Siderotech assay in order to measure which cells have increased iron binding capabilities of ENT, and are they extracting those resources from neighboring cells and inducing iron starving conditions for them. We're simply following the Siderotek assay, and the absorbance and the detection levels are at 630 nanometers, and they use a spectrophotometer for this from thermoassay. As the whole point of this part of this paper is to determine which cells are holding on and kind of clinging to that iron, as well as ruining it for other neighboring cells in which they do not have access to this necessary material. And this is where the author really emphasizes that iron, especially in the ferric 3 form, is necessary for the growth and flourishing of these cells within the gut microbiome. Figure 6 examines why E. coli's growth is promoted in the presence of ENT. And the authors hypothesize that it's because of its iron scavenging abilities. So they wanted to isolate E. coli strands that were being grown in excess within the representative gut microbiome strands, in which this was accomplished by isolating from the fecal samples, incubating in the medium we discussed previous for 48 hours on anaerobic conditions, and then restreaking for purity, of course, as well as using colony PCR via 16S rNA primers, and then being lysed with nuclease-free water. Then normal PCR amplification occurs to grow these desired strains in excess, and in turn that we can then do PCR product analysis, and we're going to do that with gel electrophoresis, specifically looking for genes that synthesize and encode for cytophores, because that is speculated to bind to iron 3 with an extra high affinity, as well as looking for enterobactin synthesis. These two factors, the authors speculate, are highly important for the E. coli outcompeting for resources within the gut, and then consequently having a disproportionate abundance relative to the other strands present. Now that we have a firm understanding of how the authors conducted their research, it's important to go into the results, analyze them, and see what the figures say based on the analyses that they've ran. Figure 1C and D represent the 16S rRNA gene amplicon sequencing data. And on the left, we have the relative abundance levels, while we have on the right, the absolute abundance levels. And as you can see, we have two controls, high, low, ETN, low, high, LOX. For the most part, all of the data looks relatively normal, except then you see a disproportionate amount of E. shigella impacted by ENT high, which this is the E. shigella is only impacted by ENT high. Conversely, the absolute abundances are quite different for ENT high. We notice that there's simply less bacteria present, and this is supporting the idea that ENT high has the most dramatic effects on the gut microbiome. What we can conclude from this is that there's massive shifts in alpha diversity, and we see that in the genre Bacteroides or Clostridium sensostricta 1, these are both impacted by ENT high and LOX high. And all these effects are drug specific, as seen by the impact on E. shigella. But we see most importantly that with taking these drugs, you're going to get a significant decrease in comparison. In a comparison to the controls, especially experiencing ENT high. Figure 1E and F is the most comprehensive figure in this whole entire paper. On the left, we have the heat map displaying the transformed abundances of the ASVs only, while figure F shows the corresponding mags. And figure F is particularly interesting because it shows that the metagenome assembled genomes, when compared to the ASVs containing 16S rRNA allow us to track and follow the drug shifts in order for us to understand what is shifting and which drugs have the most pronounced effect. Based on this data, we can tell that ETN high has the most pronounced effect in comparison to ETN low and LOX 1 and 2, shifting all with the greatest microbial shifts. Figure 2 Figure 2 represents the single-cell metabolic analysis that was that was acquired based on the SRS and FISH data. As you see on Figure D, it is the log change, full change in microbial activity. As you can see, that LOX high again and LOX low as well have similar results. E. coli is presenting more and more microbial activity as you go on, but then you can see that in Bacteroides uniformis, very comparable values to LOX low and LOX high. Conversely, this is the analysis of with the absolute abundance in the same taxa. This was at the 6-hour point, and they completed statistical analysis as well. The largest changes in microbial analysis, metabolic analysis, will be in this particular strand, while there's very, very minimal activity going on in E. coli in this particular assay. Figure 3 represents the photothermal imaging used to determine if ENT is being bioaccumulated in certain cells. So as you look onto your right, we understand that there's different signal intensities, so the green being where bioaccumulation would occur. In the case of no ETN, ENT, there is no bioaccumulation present, there's no signal, but we have in ENT high large amounts of bioaccumulation, and this is seen in both the SRS and FISH data as well as confirmed by the photothermal signal data. Looking to our right, we see figure F, which shows that E. coli specifically is accumulating a ton of ENT, and this is seen in different strands as well, and there's a lot of accumulation in this particular strand and rumi as well. And as you can see, based on the controls, there's no green, so this is confirmatory that these drugs are bioaccumulating ENT, which could be responsible for the large shifts as well as a lot of decrease in activity, and also including a lot of cell death. This could be a reason why the absolute abundance of certain certain bacteria was a lot lower in the beginning and 6-hour marks. Figure 4 is particularly important because this is where the authors and researchers kind of bring all the ideas together. So at this point in the paper, you have a firm understanding that ENT and LOX have dramatic impacts on microbial shifts in terms of composition. So what's the solution? We know that it's being accumulated, and we know that ENT has the effect of decreasing the amount of iron surrounding these cells. So can we fix the damage done to the composition by supplementing the right amount, and in a clinical sense, the right amount of iron to hopefully disregard the effects of all the iron chelation and complexing going on? As you can see, this does benefit a little bit in terms of we have ENTI high still having a lot of that E. Shigella disproportionately, but for the rest of them, there are general shifts in terms of the composition and the relative abundance towards that no drug and control. So as for example, when we have ENT high, this was the no iron right there. So when we add the iron supplement, the disproportionate E. Shigella composition is minimized to just this, which is a massive breakthrough. And that makes me question, can this be used in a clinical sense of, can we give this patients in combination with their ENT therapies. It's looking less pronounced, the effects of ENT and its particular nuances. As you can see, it's starting to mirror what no drug and iron, as well as ENT high, again, with a lesser dosage of iron. Um, this is a pivotal point in the paper because it's addressing an issue as well as a potential clinical solution. And as we move on to the absolute abundance, these changes are reflected. Um, we have no drug, and there's still a plenty of total bacteria present in the solution. We have the no drug comparison with the ENT high, and these are relatively comparable, especially considering how pronounced the effects of ENT are on many of these bacteria, that this could be a great potential clinical solution. Lastly, the authors observe in figure C that E. coli is disproportionately affected in comparison to other bacteria. We see a positive correlation with E. coli in comparison with ENT. So you have wild type E. coli as well as two variations, and throughout no drug, so this will be our point of standard. Wild type is grown significantly, and these are also experiencing growth when in comparison other bacteria are experiencing growth inhibition. So our question is, why is E. coli being out of all the bacteria not inhibited by ENT high, which has very, very dramatic and dangerous effects? And which leads us to understand that enterobactin, as well as the cytopores, are being synthesized, and it has these proper genes to chelate the ENT as well as scavenge the iron around it and use that, because we know that iron is very important for bacterial growth, but now it's taking away from all the iron from the neighboring ones, as well as having, and these are all under the umbrella term of antimicrobial resistant genes, and it increases the virulence of these particular bacteria. So this must be examined in order to understand how is this potentially going to cause opportunistic infections, and this further needs to be understood. Now I'd like to move on to further applications and how this paper is going to lead to more and more research. At this point, we need to also consider that the data and shifts have only occurred in a 6 or 24-hour span. So what the authors suggested that we need to understand a long-term study, analyzing people who haven't taken ENT or LOX, and look at their gut microbiome, and then compare it to maybe 6 months throughout the treatment. This could also be a really great opportunity to test out prescribing iron supplements alongside ENT to understand if it has a clinical benefit, because these are all occurring in vivo experiments or just within cells. But there is also raises the question of, we need to examine the effects of other antipsychotic and nervous system targeting drugs and see if they're impacting the gut microbiome. For example, depression is a increasingly hot topic in terms of how the gut microbiome impacts that, and we need to understand, are symptoms worsening? As well as exploring further mechanisms that can lead to adverse microbial effects that other blockbuster drugs may have, similar to that landmark paper in the beginning. After reading this paper, I was led with three main questions. Does ENT increase the risk of intestinal infections because you have disproportionate E. coli, as well as antimicrobial resistant genes being encoded, as well as just a very susceptible microbiome within your gut? At what point do doctors start seriously considering prescribing iron supplements for ENT patients? And is there a clinical connection in terms of the symptoms to these large microbial shifts? So are they worsening patients' symptoms of schizophrenia or Parkinson's disease? And how can we determine that? Now I'd like to move on to the conclusions of the paper and what we can discuss. The main takeaways are ENT has the largest impact on microbial abundance in the gut microbiome. While LOX has a impact, it's not as simply as pronounced as ENT. Microbial activity on the single cell level is significantly impacted, especially by ENT again, and ENT leads to E. coli and other bacteria uptaking large amounts of iron, chelating iron, and complexing iron, um, ruining the amount of resources for other cells. The microbiome the gut microbiome can be potentially benefited from iron supplementation, as we saw in figure 5. And ENT promotes iron starvation genes as well as AMR genes and increasing the potential of virulence in the gut microbiome. These are my sources, and I look forward to your discussions and comments. Thank you for your time and listening.